Hi everyone, my name's Dave Madge and I'm here to tell you about the immuno-oncology platform we've built at Biotech. Biotech is a global company with nearly 30 sites around the world and more than 20,000 employees. Our operating facilities are in China, America, Europe and Israel, and I'm talking to you from Cambridge in the UK. Today we support about 4,000 diverse organizations, from small teams in academia through to multinational pharmaceutical companies everywhere in the world. We work across all modalities and all therapeutic areas, but this discussion will focus on our immuno-oncology platforms. Our philosophy is to build open access enabling platforms that support every organization to enable their drug discovery ambitions. We work from the early stages of target identification through enabling activities such as generating proteins and cell lines, into diverse hit finding platforms, and on into drug discovery through lead optimization, toxicology, and manufacturing. Our platforms support small molecule, peptide, oligonucleotide, antibody, and cell and gene therapy programs throughout these different stages. Our immuno-oncology team now numbers more than 400 scientists, supporting target ID and validation, in vitro biology, in vivo biology, and biomarker development, including support to clinical development programs. We have substantial capacity for carrying out these studies and have supported more than 75 IND packages to date. Our tumor models are heavily annotated with growth curve, standard of care, and sequencing data. Let's take a look at some of these capabilities in more detail. In the following slides, I'd like to describe our target ID platform using gene editing tools, our in vitro immunology assay platform, our in vivo oncology platform, and translational studies using biomarkers. Our gene editing teams support target identification and target validation activities using in vitro and in vivo screening tools on both cancer and immune cells. This includes assays for drug resistance and synthetic lethality. We can create knockouts and knock-in cell lines and genetically modified mice for target validation and to support drug discovery, as well as carrying out phenotypic profiling, proliferation, and cell cycle assays. Our immunology assay platform encompasses all cell types with endpoints such as T-cell exhaustion, mixed lymphocyte reaction, antibody-dependent cellular toxicity, as well as analysis of myeloid-derived suppressor cells. Our humanized models represent all major tumor types, usually with extensive supportive sequencing and lymphocyte data, and have been used to investigate tumor growth inhibition, including tumor rechallenge and measurement of tumor infiltrating lymphocytes. We've collected extensive standard of care data with checkpoint inhibitors alone, alongside each other, and alongside other drug classes and treatment modalities. To help understand mechanism of action and to support biomarker discovery, we've developed profiling platforms for cell sorting, immunohistochemistry, macrophage migration, inhibitory factors, and RNA sequencing. We also use nanostring technology to help identify gene-disease associations in both oncology and immunology studies. To help us support antibody drug discovery programs, we've validated the use of human PBMC for adoptive tumor transfer in mice, showing, for example, that functional T cells in these models release human cytokines. To date, we've developed 56 models using human PBMC adoptive transfer, including both CDX and PDX models covering all the major tumor types and targets. To the right of this slide, you can see that we've also been using knock-in mice to express human target genes for checkpoint targeting antibodies, and demonstrating, for example, that CTLA-4 and PD-1 antibodies show substantial reduction in tumor volume in such models. Again, we've covered the major IO target genes and have built double and triple knock-in models, as well as paired Syngeneic models in both C57 and Bulb-C backgrounds. To further extend the range of modalities we've characterized, I show here some data generated using bispecific T-cell engagers in the human PBMC adoptive transfer model. In this case, we're using an immunodeficient mouse model and looking at tumor growth using a bioluminescence readout under the IVIS system. You can see that the BITE compound Blincyto provides a robust inhibition of tumor growth, whilst 2 further test substances are much less robust in their effects. To keep extending the range of modalities that we work with, I show here some data on cancer vaccine, supporting cancer vaccine discovery and characterization using the enzyme-linked Immunospot platform with, for example, tumor volume and cytokine measurement endpoints. To support clinical biomarker use, we have a CAP-certified and GCP-compliant platform for tumor tissue processing, clinical pathology, and FACS analysis. We're using the Halo digital pathology platform, and to date have validated more than 100 biomarkers. To give a few examples here, we've been working with Roche Molecular Diagnostics to support PD-L1 and mismatch repair studies by immunohistochemistry, as well as macrophage migration, this on the Ventana platform. We're also detecting c-Met alterations, such as the exon 14 skipping mutation by IHC, FISH, and BaseScope. Of course, this has been a rapid snapshot of discovery enabling capabilities, and I'd like to briefly finish by mentioning that these technologies are backed up by a comprehensive toxicology and manufacturing platform. This platform is integrated to provide IND enabling packages to all the major authorities and backed up by a strong track record of performance, quality, and timeliness. Indeed, our state-of-the-art manufacturing capabilities in China and the US have supported nearly 100 approvals for branded drugs and survived numerous regulatory body and audit clients. In addition, we recently became the first CDMO to support the approval of an innovative new drug in China under the Marketing Authorization Holder, or MAH, pilot program. I'll finish here. Thank you very much for your attention, and please do feel to reach out with questions or to request additional information.